Well, our next speaker is Christopher Lynn. Christopher got his PhD in 2020 in physics and astronomy at the UPenn, studying the statistical mechanics of human behavior. And since 2020, he has been a James McDonald postdoc fellow with Stephanie Palmer, David Schwab, and Bill Bialek. And he's going to talk to us about statistical physics of emergence and dynamics in neural systems. Hopefully.
Yeah, okay, we're good. Okay. Yes, thank you.
I'll let you know.
Okay, cool. Yeah, thank you for the update. Can y'all hear me in the back? Yeah, everything's good. Okay, yeah, thank you for the introduction. I'm Chris. I'm happy to be here today to talk to y'all about some research that I've been thinking about having to do with the statistical physics of the brain, sort of broadly speaking. So in my research, at the broadest scale, the question that drives the things I think about are what drives neural function. And to give you a sense of why I think this is such an interesting question, inside each of our heads, we have 80 billion neurons that are interconnected in this complex web of connections. And these interactions somehow build upon one another to generate macroscopic phenomena that we're used to, like cognition, planning, information processing. But there's sort of a large scale question, which is how can we distill all of this complexity to some organizing principles so that we can sort of generate knowledge? And to start to think about that, what I like to do is I like to start with simple and interpretable hypotheses, ideally hypotheses you could state in plain English. And at the end of the day, I try to build quantitative models and frameworks that you can then finally at the end compare against real data. And the key bridge between these two is using ideas and methods from StatMech and information theory and network science. So I'd like to sort of demonstrate this process for you a couple of times. And what we'll see is that sometimes the insights that we gained aren't just applicable to the brain, but you can actually think about other sort of collective phenomena in biology. So I won't have time to talk about all my research, but I just wanted to sort of give you a sense of my trajectory. So this is, let's go here again. So this is time in sort of arbitrary units of my life. This is scale in real physical units of meters in terms of the system that I was thinking about at the time. As you can see, we're sort of trending down. So maybe by the time I get here, I'll be doing sort of quantum mechanics or something. So to start my graduate school, I was interested in thinking about the principles of inference and control in social networks. And then the bulk of my graduate research had to do with human cognition. So not yet looking inside the brain, but using sort of large scale cognitive experiments and bridging that with information theory to try to quantify how much information are people processing from a given stimulus. Then this was a sort of project at the beginning of my postdoc where we used information theory, specifically rate distortion theory, to try to quantify how compressible is a complex network. This is a project that I'm particularly proud of that we finished recently. So this was led by Aaron Tyke, who's a new professor at Wellesley, looking at sort of gender bias in physics citations. So the sort of bulk of my postdoc research has to do with thinking about irreversibility or the non-equilibrium dynamics in sort of complex living systems. And sort of currently and moving into the future, some things that I'm thinking about have to do with sort of the statistical physics of these new large-scale measurements that are coming out. So I want to talk to you about sort of these two last sort of groups of projects. I'm not going to talk about all of them. But to start, I'd really like for us to think a bit about how can we think about irreversibility or the arrow of time in a system emerging from interactions in some sort of complex system, like the brain, as an example. So in biology generally, so this isn't just applicable to the brain, in biology generally, we have this sort of notion that you start with the burning of energy, and somehow that is driving biological functions. Like in the context of the brain, you can think of cognition, sensing, information processing, but you could also think much more broadly about whatever your favorite biological function is. And now the key sort of connection here is irreversibility. You burn energy in order to maintain yourself away from equilibrium in order to perform directed functions that are directed in time. And you're going to hear me talk about irreversibility, non-equilibrium dynamics, the arrow of time, and I'll make these sort of connections more precise in a moment. Also, if anyone has any questions at any point, just feel free to interrupt me. So these links between non-equilibrium dynamics and biology are becoming more and more sort of well understood as we get better and better biological measurements. So dating all the way back to Hopfield in the 70s, we had this notion that non-equilibrium dynamics are absolutely vital for kinetic proofreading. We now know that non-equilibrium dynamics are necessary for computations and adaptation at the cellular level. So this is a recent paper with Ben and Mike Morrell looking at the irreversibility of sort of biochemical oscillators. And this is also a paper that I did in collaboration with my graduate advisor, Danny Bassett at Penn, looking at the relationship between irreversibility and cognition in the human brain. And we'll talk about this a little bit later. But these links between irreversibility and sort of biological functions raise a sort of clear question, which I think is very well summed up by this quote from Schrödinger back in the 50s, so way back then. And he says, how would we express in terms of the statistical theory the marvelous faculty of a living organism by which it delays the decay in the thermodynamical equilibrium, which in the next sentence he equates with death. So to paraphrase, to parse this, he's basically asking, how can we use statistical physics to understand the inherently non-equilibrium nature of living systems? Now, unfortunately for Schrödinger, we didn't really have a good statistical theory of non-equilibrium physics at the time. But over the last 20 years, we've really been building up these notions of a statistical physics of non-equilibrium systems, a sort of field known as stochastic thermodynamics. So the key insight from stochastic thermodynamics is a relationship between two sort of on the surface seemingly unrelated types of quantities. One that has to do with the production of entropy in the universe. So this is the physical rate at which the entropy of the universe as a whole is increasing due to some process or some system maintaining itself. Now, in certain circumstances, for example, if the system is connected to a heat bath, if it's at steady state, you can think of this increase in entropy as related to sort of the dissipation of heat or the amount of energy that a system needs to expend to keep itself away from equilibrium. Now, the key insight from stochastic thermodynamics is that this purely physical notion of the arrow of time is related to a purely information theoretic notion of the arrow of time. So I'm assuming here that the fundamental laws of physics are Markovian. So here, what we're talking about, just to define some things, is this probability from x to x prime is the probability of being at some state x at time t, and then going to some other state x prime at the next time step. And I'm going to refer to this information theoretic quantity as I dot, not to be confused with S dot, the physical production of entropy. Now, what this quantity is a KL divergence between these forward time trajectories and their reverse time trajectories. And if you're not familiar with KL divergences, you can just simply think of it as a difference between two probability distributions. So two main things to have in mind are that this quantity is zero if these two distributions are exactly equal. So forward in time and backwards in time dynamics are the same. And it can only be non-zero if they're any different. So it's like a difference. Another sort of interesting aspect of this quantity is that, and this is sort of a nice thing generally from information theory, so you can often convert things with logs into things in the English language. So literally what this quantity represents is the evidence that one time step conveys about the arrow of time, about the difference between forward in time and backwards in time. So it's a purely information theoretic quantity that we will go estimate in bits. So just to give you a sort of a intuition for what a reversible versus an irreversible system looks like, here we have, for example, a reversible system. So these nodes represent states, so x and x prime. The arrows here are meant to represent these transition probabilities. And now if we obey detailed balance, that's simply the statement that these transition probabilities are equal to their time reverse. And we know from what we just talked about with KL divergences that this KL divergence will be zero and there's no irreversibility in the system. It's an equilibrium system. And also, just to give you, you can already intuitively see why there's no arrow of time in this system. Because if I played you a video of this system and then I played you that video backwards, you statistically would not be able to tell the difference. There would be no difference between the two. Now you can contrast this with irreversible dynamics. So when detailed balance is broken, that's simply the statement that the probability from x to x prime is not equal to its time reverse. And then you end up with these net fluxes. So if x to x prime is bigger than x prime to x, and you end up with these net fluxes, which in steady state loop back on themselves like this. So this is sort of a characteristic feature of steady state non-equilibrium systems. And you can see that because this is a difference, as we talked about, any sort of flux, any breaking of detailed balance will increase this irreversibility, this KL divergence. And in terms of the arrow of time, if I played you this video forward, you would see more often than not the system sort of going this way. And then if I played it for you backwards, you'd see movement in the counterclockwise direction. So you would clearly be able to detect the arrow of time here. Now this sort of brings us to the question that I wanted to start with, which is in stochastic thermodynamics. So usually when we're thinking about these systems, it's typical to sort of think of one state variable x, and it's moving from x to x prime, and there's some network of states, just as I've sort of illustrated here. But in biology, things are a bit more complicated. Almost always, we're interested in systems where there's multiple degrees of freedom. There isn't just one state variable. There's multiple independent, or they might not be independent, but multiple degrees of freedom, possibly interacting with one another to generate these sort of macroscopic collective patterns of activity. And the question then becomes, if we sort of think about the irreversibility of the population, of the whole system, how can we tell where this irreversibility is coming from? Is it coming from, for example, independent elements? So if the elements were all non-interacting, would you still have the same irreversibility? Or is it coming from sort of pairwise interactions between these elements, and so on and so forth, up to sort of the most complicated possible interaction, which is a full n-body interaction? And I want to be clear, I have to keep hopping between, this is one unfortunate thing about stochastic thermodynamics, you have to hop between state networks and degrees of freedom networks. So here, these nodes represent individual degrees of freedom, like neurons or bacteria interacting, or animals in a population. Okay, so we want to know how could we ever tell if the collective irreversibility is coming from one of these types of interactions or independent dynamics? So what we can do is we can write down the dynamics that fully define these sort of interactions of different orders. So all you need to know to define the independent dynamics is just the individual sort of transition statistics of the individual elements. To define these pairwise interactions, you just need to know sort of how the pairs of states behave together. I mean, the pairs of variables behave together, and that's for all pairs. All the way up to the point where, when we're talking about an n-body interaction, to fully understand what's an n-body interaction, you need to just fully define the dynamics of the system. So you have the full state variable x going to some other state variable x prime. Now, what we can do is we can define the minimum irreversibility consistent with some order of dynamics. And so here, for example, we have the minimum irreversibility consistent with independent dynamics. So if I told you the independent dynamics of the system, this is what you would say, this is the minimally irreversible, sort of the maximally equilibrium system that you could write down. Now, interestingly, these minimum irreversibilities form a hierarchy, and there's a lot of information contained here. And the statement is simply that if I give you information about larger groups in the system, you necessarily have to have a higher minimum irreversibility because I've given you more, I've constrained your system more. And so just to give you a sort of intuition for what's going on here, let's focus on this end of the hierarchy here. If I gave you literally zero information about the system, I just said, there's a system in this box, tell me something about its irreversibility. Simply because it's a KL divergence, you already know that it has to be bigger than or equal to 0. And this is just a mathematical restatement of the second law of thermodynamics. Now, I could tell you about the individual elements behaving, and you would get more information about the irreversibility, and pairwise interactions, more information, and so on and so forth, until if I tell you about the n-body interactions, these full dynamics of the system, then we've just recovered the full irreversibility of the system because you've told me all of the dynamics. Now, really what we are interested in is how much irreversibility arises just due to some order of interactions. And to quantify this, we look at the difference between one order and the one below. So this is the extra irreversibility you get from having kth order interactions that you didn't have from just having k minus 1th order interactions. And we call this the kth order interaction irreversibility. And we know from this hierarchy that this has to be non-negative. So now we've sort of set out to get exactly what we wanted to at the beginning. We have this full decomposition of the irreversibility of the full interacting system into these different non-negative components. So the independent dynamics, the pairwise interactions, and so on and so forth. And now what you can do, and I want to sort of emphasize, this is a purely physical framework. I haven't said anything about brains. I haven't said anything even about biology. You could apply this to any system with interacting elements and ask, where is this irreversibility coming from? But as I sort of alluded to, we are interested in brains here. So that raises a question: why would I be interested in thinking about irreversibility in the context of neural dynamics? So the reason is sort of twofold. So first, we talked about these 80 billion neurons inside your skull. They make up roughly 2% of your body mass, but it accounts for roughly a full quarter of your body's energy expenditure. And it's also capable of all of these interesting sort of cognitive functions that we talked about. So it might be a rich sort of playground to think about irreversibility. And a second point from a more sort of philosophical bent is that our perception of the arrow of time is purely due to whatever dynamics are happening inside our head. So the fact that we remember the past and we plan for the future and not the other way around has to do something with what's going on inside our head. And not only that, what we really started focusing on is retinal cells. And the key insight here is that the only information that your brain has about the outside world visually has to be passed through the channel of your retina. So any sense in which you say that movie looks like it's moving forward in time and not backwards in time, that information has to somehow be encoded on the retina and passed to the brain. So we're going to start with thinking about the retina as a sort of an interesting system. So this is data collected by Olivier Marr and Michael Berry. And so here we have sort of a picture of the experiment. So these are neurons, and they place this sort of high density electrode array indicated by this square here. And then you can record the electrical activity of these retinal neurons in a salamander retina while it's being shown this visual stimulus. So here to start, we'll think about a natural movie. So for a salamander, a natural type movie might be fish swimming in a stream. So that's what they're sort of seeing here. Now, what they can do is they can deconvolve the individual spikes in these neurons. And so here what I'm showing you is on the y-axis, the cell number, and here time in the experiment. Each dot here represents one spike from one neuron. And I want to point out that these neurons naturally form binary states, 1 for spiking, 0 for not spiking. And so what we can do is we need a collective state of the system. So what we do is we can define a time window, here 20 milliseconds, which is sort of typical. And you can look at the entire activity of the system within that window, and you get a full state vector, a full binary state vector, 1 or 0 for each neuron. And then as we slide this window forward in time, what you get is a dynamics of the system, the collective dynamics moving from one state to the next. And we can ask, are these collective dynamics defining an arrow of time? Is there irreversibility in these collective dynamics? Now, the first thing you could think to do is just look qualitative visually. And to do that, we're going to focus on 3 neurons because you can't visualize anything higher than 3-dimensional. So here we have sort of 3 neurons. Now I'm converting back to these nodes represent states. So these 8 states are the 8 possible binary combinations of 3 variables. So all the combinations of 3 zeros and 1s. And what I'm plotting in terms of the arrows are the net fluxes in units of per time from one state to another. And what you see is almost exactly like the cartoon I drew earlier, if you look in the back here. So you see this sort of loop of flux happening in state space. And indeed, we do find that actually the neurons are in steady state. So any breaking of detailed balance has to loop back on itself like this. So this is cool. So we visualized the breaking of detailed balance in this system, but can we actually quantify the irreversibility, this KL divergence? And I want to kind of point out that estimating KL divergences from data is quite a tricky subject, especially in high dimensional data. And so we had to be very careful to make sure that we were estimating things accurately. And it turns out that for the largest sort of group of neurons that we could look at is 5 neurons. We would love to go to more, but we didn't want to go beyond what we knew that we could actually estimate accurately. So for random groups of 5 neurons in this population, what we can do is estimate this irreversibility. And it turns out, so over 100 random groups, we find that roughly 2/3 have significant irreversibility. 2/3 define a significant arrow of time, whereas 1/3 look as if they're statistically in equilibrium with values here. I mean, so the ones that aren't, that have significant irreversibility are plotted in this histogram. So I want to point something out. So now we could ask interesting questions. Now we built up this framework to think about irreversibility in these systems. You could ask, well, what is the role of the stimulus on the retina? And as we've talked about, so far, we've only considered this natural stimulus, which is fish swimming forward. And if I played you this video, I decided not to do it because videos are always tricky. But if I played it for you, you'll just have to trust me that if I played it backwards in time, you would see a fish swimming backwards and be like, that's an arrow of time. There's something clearly different going on there. But now we can show the exact same neurons at different stimulus that by definition is totally reversible in time. It's a fully equilibrium dynamics. So this is just a horizontal bar, sort of stochastically moving up and down, defined by Brownian motion, fully reversible system. You wouldn't be able to tell the difference between forward and backwards. Now we can go back into the retina and ask, is there still irreversibility in the retina, even if there's no irreversibility in the stimulus? And not only is there still irreversibility, the irreversibility increases, which is something that makes no sense to us. I mean, the only statement we can make here is that it is not simply the case that the irreversibility of the visual world is imprinting itself on the retina. Something else is going on, something about the computation that the retina is having to perform. So now that we've sort of built up the framework for actually being able to estimate these things in sort of this real data, we can go back to our framework and ask, where is this irreversibility coming from? So what I'm plotting here is on the x-axis, we have the order of interactions k, so 1 is independent. And on the y-axis, we have the fraction of the total irreversibility consistent with the kth order interactions. Now, one thing that might be extremely confusing is what is going on down here. Why are independent neurons not producing any irreversibility? And it comes from this interesting sort of mathematical fact that if you have a binary steady state system, which our neurons are, and we're only looking at sort of Markovian dynamics, the sort of local information about the arrow of time, in systems that define that way, an individual binary variable cannot define an arrow of time. And to help sort of guide your intuition, you only have 2 states, 0 and 1. And if you think of a long string of 1s and 0s, in the long run, the number of times you go from 0 to 1 has to be the same number of times you go from 1 back to 0. You can't keep going from 1 back to 0 many times and not have gone back from 0 to 1. So there you can have, by definition, no net flux from one state to another. So a single neuron in this system cannot define an arrow of time. And what this means is that this irreversibility that we're measuring is purely a collective phenomenon. So you have to have at least 2 neurons to define an arrow of time after you binarize them and look at the Markovian dynamics. And so what we find here is that almost 70% across both stimuli, roughly 70% of the full irreversibility of these neurons is captured by pairwise interactions, which again are the simplest possible source of this irreversibility. So just to sort of conclude this part of the research, we've shown that neurons can collectively define an arrow of time, even if the stimulus doesn't. And this collective irreversibility tends to come from the simplest possible source, which is pairwise interactions. And that gives us hope that we could come up with some sort of network models of how these neurons are interacting with one another to generate this non-equilibrium dynamics. So before moving on to other ideas, I'd quickly like to talk about sort of ways I see this moving forward. So we have this decomposition we've written down. I mentioned that there's nothing particular about the brain that this framework relies upon. So we could immediately go out and ask similar questions in other collective systems like active matter systems, interactions in animal behavior, or even within a single animal, interactions between different parts of its body. And also sort of interactions at a larger scale in the brain between different brain regions, which is something I'm going to talk about in just now. So I threw up this sort of illustration before where we have this notion of burning energy supporting biological functions via irreversibility. So far, we've just focused in on the irreversibility part, and we've asked about questions about the nature of irreversibility. How does it emerge from interactions in the system? But there's also different parts of this sort of cartoon that you could focus on. So for example, how is irreversibility related to biological functions in the brain? And this is precisely a question that I set out to think about with Danny Bassett at Penn. And what we did is we looked at human neuroimaging data. So this is human brains people sitting inside an fMRI scanner, and you can record the collective dynamics of each brain region over a period of time. And not only that, you can do it while the human is being asked to do something different. So they could just be resting in the scanner, or they could be doing some sort of more cognitively demanding task. And so as we've talked about, you can estimate this irreversibility. And this is in 600 subjects that we didn't gather, by the way. This is, we did not take the time to get 600 subjects. This is an openly available data set. And what you see is that when people are sitting in the scanner being asked to do nothing, almost always they just fall asleep. You see that the brain is statistically, at least at these large scales, very close to equilibrium. There's almost, you cannot tell the arrow of time, almost. You can tell a little bit, but it's very small relative to errors. Now, as we move forward in these different types of cognitive functions, what we see is that the irreversibility, the large scale irreversibility of the brain increases by a lot, by tenfold. So if you're doing a gambling task where you have to remember what cards you're looking at, or a motor task where you're only being asked to like wiggle your left finger, wiggle your tongue, the irreversibility of the brain jumps up by roughly tenfold. What this means is that the brain is purposefully pushing itself through these macroscopic states that you can visualize. That I'm happy to show people these networks of flow between these large states. It's really interesting. And this brings me to something that we haven't done that I think would be a really cool thing to do, which is go back to this cartoon over here and look at the link between energy and irreversibility. So in these same MRI scanners, another thing they can do, so in addition to thinking about the irreversibility of the brain, you can also measure you can get, well, not directly, but you can get pretty good estimates of the metabolism going on in different parts of the brain with relatively good precision. And therefore, you can directly start to think about links between large-scale metabolism in the brain and irreversibility, which then, as we've talked about, we might think is related to cognitive function. Okay, so that sort of wraps up us talking about irreversibility for now. So we've talked about how can we use these new non-equilibrium theoretical ideas to study the brain, but I'd also like to think about from a more traditional equilibrium statistical perspective, how can we use more standard techniques to think about these extremely large-scale neural experiments that are coming out right now. So this again is our sort of driving question: what drives neural function? And on the experimental side, at least at large scales in the brain, there's been a lot of recent progress. When I say recent, I mean like over the last 3 years. So for example, people can now image from 1 million neurons simultaneously using calcium imaging. And just to, again, sense of scale, I think, is important here. So this is in a mouse. Mice have roughly 70 million neurons. So this is more than 1% of the entire brain of a mouse you can record. So you're now starting to think about sort of macroscopic fractions of the brain. Now using electrophysiological sort of recordings, which have much better temporal resolution, by the way, you can start to look at tens of thousands of neurons. And also for lengths of over a month. This kind of blew my mind. So they stick these sort of probes, these electrical probes inside the mouse's head. The mouse can run around for a month. You continuously record the electrical activity of 10,000 neurons over the span of a month, which is kind of blows my mind. On the structural side, not only can we think about these large-scale neuronal patterns of activity, but you can also start to map out the physical connections between neurons. And so this is a data set that we're going to talk about in a second. So this is out of Genelia, and they've mapped out roughly a full quarter of the fly brain, so 22,000 neurons. Now, in addition to these efforts at the neuronal level, there's also sort of automated tracking has led to a revolution in animal behavior. So this is a paper by Thierry Emmenet here and Damon Clark. And also, I think an underutilized resource is actually online human cognitive experiments. So this is a project that I led in my graduate research, where we were able to gather data from 400 human subjects within a week, because you can put it up online and you have to be careful about making sure they're paying attention, but you can gather an extremely large amount of information really quickly, experimental data. And so all of these sort of large scale experiments raise a question, which is, what are the links between these fine scale interactions, and how do they build upon one another to give rise to these macroscopic properties? And this sort of abstractly is precisely what statistical physics was sort of built to do. Think about, how do you start with microscopic mechanisms, and how do you derive macroscopic predictions about behaviors at the large scale? So I want to give you just sort of two examples of directions that I think are interesting here. So focusing on large scale neuronal activity, these recordings of like 1 million neurons, let's say. Suppose you wanted to understand how, if I started with sort of fine scale sort of measurements of my system. So for example, suppose we only measure the average activities of the neurons and the pairwise correlations, let's say. How could I know what macroscopic features of the system are predicted by these fine scale measurements? So for example, is it just the case that I only need to know these quantities to predict the full state, the collective sort of distribution over states, or the chances of many neurons firing together, or actually maybe even the structure of the physical interactions between neurons? It turns out the most unbiased way of making this map from measurements to predictions is precisely the maximum entry principle. Now, anyone that has worked or thought about the maximum entry principle understands that there's sort of a fundamental computational bottleneck. And even for those that haven't worked with the maxent principle, you can still gain an intuition. So basically, if we're thinking about average activities and correlations, and our states are binary, which is almost always true for networks of neurons, then the maxent model is exactly, not analogously, it's exactly the Ising model. And so you know that if you ever try to compute a correlation in an Ising model, you need to run a Monte Carlo of any reasonable size. You need to run a Monte Carlo simulation. And it's even worse because we're doing the inverse Ising problem. So you need to run many Monte Carlo simulations. And it turns out that, roughly speaking, you can use these methods to study populations of about 100 neurons. That's roughly the state of the art. This raises an extremely clear question. How can we scale these principled statistical physics models up to 1 million neurons? We're orders of magnitude off here from state of the art to the actual experiments being run now in labs. And I won't go into the details of how we're going about solving this problem, but this is work with Bill Bialik and Stephanie Palmer, and we've constructed a method that basically the key insight is, instead of looking at all the correlations, focus on very specifically chosen correlations between neurons such that they give you the maximum possible information about the population as a whole. And it turns out that you can do this in a way that scales. I have examples on my laptop that you can scale it all the way up to 10,000 neurons in 30 seconds on your laptop. So that was neuronal activity. So what if we thought about neural function? I mean neuronal structure. So as we discussed a second ago, there's this really cool data set from Janelia looking at the physical connections between neurons in the fly brain. So I'm sort of plotting you a little bit of it here. So there's roughly 22,000 neurons. There's over 3 million connections. And here I'm only showing you the top 0.1% of the connections. If I showed you all of them, it would just be a big blue blob. So I had to sparsify it. Now, we started looking at this data just because we thought it was so cool, and started thinking about the network properties of these physical connections between neurons. And one thing jumped out to us immediately that totally blew our minds, which is this distribution. So let me describe what we're looking at. So what we're looking at on the x-axis is the number of synapses in one connection, one pair of neurons. How many individual contacts are there between those two neurons? And you can think of this as sort of a proxy for connection strength. Now, I want to point out that the typical connection strength, the typical number of synapses, is on the order of 1. So this dot here represents what portion of connections have 1 synapse, and that is roughly 50%. That dot is 50%. And to give you another sense of scale, the average number of synapses in a connection is like 4.5. It's order 1. Yet we have this extremely heavy tail where there's some connections, an individual pair of neurons with 1,000 individual connections between those two neurons. I've studied neuroscience for a while, and I didn't even know that that was possible. And so this heavy tail in this distribution, what it represents physically is this sort of sparse backbone of extremely strong connections. It's the most important connections in the population. And for a sense of scale, if you were to sort of randomize the placement of these synapses, you would get something totally different, obviously. This is a Poisson distribution, and you wouldn't see any connection stronger than like 20 synapses. So orders of magnitude different. So clear question, where is this coming from? How could we think of a simple network mechanism where a network like this would self-organize towards looking anything like this? And the key insight, so this is work with Caroline Holmes and Stephanie Palmer at Chicago. The key insight is that there's this really interesting feedback loop in this system. So we've talked about neuronal activity, and it turns out there's a feedback loop between structure and activity in these systems. So you have the structure, the physical wiring between these neurons, that guides the correlations between the neurons and their activity on a fast time scale. So that's this arrow. You have a fast time scale in which the activity is generated. And then you have a much slower time scale in which it loops back on itself. So Hebbian, like you were asking earlier. So if two neurons are more correlated with one another, they're more likely to grow a stronger connection. So not only does structure influence activity, activity influences structure in this feedback loop. And so we wrote down what we think is roughly the simplest possible model we could think of this extremely complicated feedback loop. And it has one single parameter. And this parameter, it represents the fraction of random dynamics in the network versus these types of dynamics. That's the only parameter, which is sort of balanced. And you can fit this one parameter to this distribution. And what you find is this prediction, which we're very excited about. And not only that, I'm happy to talk with other people after this, but there's other data sets of this kind, and it looks very, very similar. And we're really excited about this. So just to conclude, we've sort of talked about my research trajectory and talked about thinking about irreversibility and how we can sort of decompose this non-equilibrium dynamics into different interactions. And we've also talked about the statistical physics of large-scale neural measurements and the insights that physics can shed on these new exciting experiments. So with that, I would just like to thank my collaborators and my funding sources, and thank you all for listening.
Thank you. Here, this. Joe.
Very interesting.
Thank
The just going back to the the irreversibility in the first half. I mean, the elephant in the room here is that the irreversibility much less than the actual entropy distribution.
Yes.
And we're not talking about small. We're talking about millions, billions of folds.
Yeah.
In the brain, it'll be billions of folds.
Yeah.
So that raises the question about how much is the brain or biological system willing to pay for information. It must be a lot. And I just wonder whether you've thought about this problem.
No, it is certainly. So what he's bringing up is the fact that when I showed that inequality before of physical entry production and this information theoretic notion, almost always when you're thinking about coarse-grained systems like what I've been describing with neurons and brains as a whole, that bound is very far apart. So the amount of energy that the brain is actually expending is much larger than the irreversibility we're able to detect at large scales. And I think it's a very interesting question. Me and Ben were talking a little bit about this yesterday. How can you start to try to talk about systems where these are maybe not on the scale, but within an order of 10 or something much more than like you're talking about. And I think the natural place to start would be like one neuron. So this is something that Ben and I were talking about. That would be, obviously, that's the simplest neuroscientific unit. It would be it's the most fruitful for thinking about the actual mapping from the thermodynamics of energy and the irreversibility. That's certainly a good question.
Hi, thank you for your talk. In your model, when you have these pairwise tertiary quaternary interactions, are they considered generally symmetric? And then I guess when you look at the data that you show with the synapsing, are they an equal number generally A to B, or is it not 750 to 250 BA, for example?
Yeah, that's a good question. So first and foremost, they are certainly not symmetric, because if they were symmetric, you would have an equilibrium system, which is something I haven't talked about. But when I was talking about the excitement about building sort of a network model of these neurons, those interactions between the neurons necessarily will have to be asymmetric to generate any non-equilibrium dynamics, to answer your first question. Secondly, when we look at the interactions, we're looking at all possible interactions. So all possible pairs of interactions, all possible triplet interactions, and so on and so forth. So we haven't yet thought about sort of sparsifying those interactions. Thanks, though.
Hello, thank you for the great talk. I had a quick question. In your attempt to go from measurements to inferring, I guess, the states, you said one of the things that you did was to identify the sparse neural networks. I was wondering exactly what methods you use to do that. Is it sort of like you use anatomical knowledge, or you use unsupervised methods, or heuristics, or?
Yeah, so we're only looking at the statistics of the activity. So what we're looking at is just the simplest thing you could do, which is just the pairwise correlations between the neurons. So is neuron I correlated with neuron J at some time? And then the key is that perhaps you could just focus on the strongest correlations in the population, and maybe that will give you a lot of information about the whole system. So we're trying to think about the sparse network of the strongest correlations rather than just looking at all correlations, which doesn't seem biologically relevant anyways, because every actual network in real life, particularly the brain, is very sparse. That's something we're excited about.
Okay, great talk. I have two questions regarding the fraction of the irreversibility. So the first question is, does the conclusion that the second order I dot always dominate, like if you like change n from 3 to 5? I know like the maximum n is 5 in this case. And the second question is, if that is presumably true for like higher number of neurons, can you estimate IDA like just by measuring the pairwise marginal stability of all the neurons?
Yeah, so to answer your first question, so all, like we said, all we have access to is 1, 2, 3, 4, 5 neurons. We can't go past that without not being sure we're estimating things correctly. Certainly in all of those relatively small groups, it's the pairwise dynamics that are dominating. Whether or not, and then to your point, we're excited to try to think about larger populations. And if it is still the case in larger populations that it's still driven by pairwise interactions, then we can, like we were talking about, build these sort of network models that we're used to thinking about in neuroscience to talk about this irreversibility. That's a good question.
Okay, last question before we need to go get coffee.
A quick one. So on your graph of the probability versus the number of connections, 2 slides ago. So can I think of probability as the fraction of neurons that are connected by a certain number of connections?
It's fraction of neuron pairs. So this is a distribution over the pairs. And it's just how many synapses are connecting just that pair of neurons.
Right. So there is a skeleton which contains only of 1 billionth of the entire number of pairs, right?
The number of pairs, well, it's roughly, I mean, this number squared. So
yeah, I understand. But I mean, this peak is around probability of around 10 to minus 7, 10 to minus 6 point.
Right. Yeah. These, you're saying?
So is it possible somehow to visualize spatially how these links are?
Yeah. So for example, if we took this network, this is almost precisely what we're looking at in this network. If I kept looking more and more sparsely, then you would start to see these are the connections that would be left. These strongest connections would be the ones that are left. And then you could actually see this extremely strong backbone of connections that's sort of driving the circuitry.
The entire square, or they are somehow...
Oh, that's a great question. Yeah, when do you get a connected component, for example? Not sure. There's so many other, this is one network property that we looked at. I've only talked, there's so much more to explore here. That's a very good question.
Okay, so we're going to bring that now to the coffee. Don't bail on me. At basically 11, I want you all here. Okay, we're exactly on time, so let's keep it on time. Thank you.